Welcome friends. We will be discussing about the MCQs related to plant science. These MCQs are very important for the ones appearing for the entrance exams related to plant science, be it BSc, be it an MSc, be it for PhD or any entrance exams related to JRF, SRF and other RET examinations. Okay, so let's begin. We will be solving several model papers. This is model paper 1. Question number 1 is the ability of a genotype to produce a relatively narrow range of phenotype in different environmental conditions. Options are adaptability, annotation, acclimatization, or gamogamy. So first of all, the genotype which can produce a narrow range of phenotype is adaptability. Because when we see about annotation, when we do DNA annotation is specifically finding the location of the gene and the coding regions, and also to know what is the function of those coding regions. This is about DNA annotation. Acclimatization is the temporary adjustment to a particular environment, and agamogamy is the term is not specific here. Homogamy, if we discuss about, it is the maturation of male and female gamete at the same time. So here for question number 1, the ability of a genotype to produce relatively narrow range of phenotype is adaptability. Next question is 1 kilogram is equivalent to. We all know this, 1 kilogram is equivalent to 2.20 pounds. The third question is the genome contained in bread, wheat are called to be as. So the genome contained in bread wheat is of A, B, D type. If you remember, AA crosses with BB and then AABB crosses with DD. So the genome contained in bread wheat is ABD. Next is yellow vein mosaic disease of okra. Okra is lady's finger. Is caused by pathogen called. So it is by Abel-Moschius esculenta. Question number 5 is dangerous gas for the depletion of ozone layer is chlorofluorocarbon. Number C. Question number 6 is the first antiseptic agent was discovered was carbolic acid. Next is foundation seed is produced from breeder seed. Next is oral gangrene is due to the loss of catalase enzyme. This catalase enzyme is mainly responsible for breaking of hydrogen peroxide to detoxify the cell. Okay, so oral gangrene is due to the loss of catalase enzyme activity in human beings. Are the symptoms of the disease is called acatalasia, because when catalase is not there, a symptom called acatalasia will be seen, and it is an autosomal recessive peroxisomal disorder where the catalase is there is the loss of catalase, as a result of which H2O2 built up in the system, and this H2O2 leads to the damage of the cells. This is a very harmful disease. Number 9, a group of plants produced from a single plant through asexual mode of reproduction. So this is clone. Okay, the genetically identical organisms will be produced when there is asexual reproduction. There is no mixing of gametes. Next is, okay, first of all I would like to let you know the Bloom syndrome also. This Bloom syndrome is what? It is the rashes which happens near the nose and near the cheek towards the nose. This is due to the sunburn. Okay. The next question is the book Fungi and Plant Diseases was written by Okay, it was written by Mundakur. Question number 11. The situation in which two different alleles for a trait are expressed unblended. That means both the alleles are co-expressed. Neither one is dominant or suppressive. Both are co-expressed. We call this phenotype as codominance. Right? Loss of chromosome segment is known as, so when there is loss, we all know it's a deletion. Next is fractional sterilization is. So what is fractional sterilization? So while knowing about tyndallization, we will know about pasteurization also, because these two are relatable terms. First of all, understand about tyndallization. What is tyndallization? That this is a sterilization method and it sterilizes all spores and vegetative cells by heating them for 100 degrees centigrade for 3 consecutive days with an incubation period in between. Okay, pasteurization is the moist heat treatment. So this pasteurization is the moist heat treatment, and this pasteurization, it eliminates the pathogenic microorganisms in milk and other beverages. Okay, by heating, cooling and sealing. So heating, what are the 3 processes involved? Heating, cooling and sealing. So this is killing the microorganism present in milk. Okay. Now for heating in tyndallization is at 100 degrees centigrade, but heating in pasteurization include either 63 degrees centigrade or 72 degrees centigrade. So if it's of 63 degrees centigrade, it will be for 30 minutes, and if it's for 72 degrees centigrade, it is for around 15 to 20 minutes. And the scientist who discovered tyndallization was John Tyndall, and the scientist who coined the term pasteurization was Louis Pasteur. So tyndallization is mainly involved in sterilizing the culture media and chemical solutions which can't be heated above 100 degrees centigrade. Okay, and tyndallization kills all the spores and vegetative cells, and minimum 3 days is required for tyndallization process. But pasteurization is a little bit faster process. It's not such a time consuming. It doesn't kill spores. Pasteurization does not kill spores. Does not kill spores, but it eliminates the pathogenic microorganism. Okay, and the method which is used in tyndallization is fractional sterilization. Okay, now questions from tyndallization and pasteurization are very common. You should remember the difference, right? Next is the adjacent cell of some fungal hypha brought into communication with each other by means of loops is called as. So the fungal cells which are connected, one fungal cell is connected to the adjacent fungal cell via a connection known as clamp connection. Okay, so this kind of connection through which the nuclei can move is known as what? This is known as clamp connection. And for example, in Basidiomycetes, clamp connection is visible. So answer will be A. Next is the Indian scientist who was awarded the Nobel Prize for DNA synthesis. When it's about Indian born scientist for DNA synthesis, it will be Hargobind Khurana. Next is number 16. Introduction of DNA into cells by exposing them for very brief period to high voltage electrical pulses, which is thought to induce transient pulse. Transient means temporary, for a short period of time, in the plasma lemma is called electroporation. That means when we need to introduce any recombinant DNA into the cell, what has to be done, that those cells has to be exposed to to the brief period of high voltage electric pulses, which will make pores in the cell membrane so that the DNA can enter, and this pore is for is these are the transient pores. That means the membrane is self-sealing. After the DNA is taken in, the pores automatically they become sealed. So this formation of pores due to electric current is electroporation. It's very important part of recombinant DNA technology. Now, the study of differences and similarities in genome structure and organization of different organisms is called. So what it will be called, that when we compare or when we study about the differences and similarities in the genome structure and organization of different organisms is comparative genomics. Okay, why genomics? Because we are comparing the genome. Number 18 is the protective sheath that covers the young shoot of the embryo in plants of the family Gramineae. So Gramineae is a monocot family of the plants, and monocot family, the young shoot is covered by a sheath or a covering. Sheath means a covering. This is said to be as coleoptile. But in place of shoot, if it would have been root, then it will be coleorhiza. Understood? Next is number 19. All bacteria having a conjugative plasmid will have a. So conjugative plasmid means there will be a tube which will be formed, which will help in the transfer of DNA from one cell to another. So this conjugative plasmid will have sex pili. Okay. Number 20 is when a disease occurs periodically in a widespread area causing devastating damage to the crop plants, it is known as. So first of all, it is periodically widespread causing devastating damage to the crop plants. What it will be? It will be epidemic disease. Okay, because it is periodically spread, occurs periodically in a widespread area. First of all, what is pandemic? Pandemic, it is, say for example, Corona is the best example. We are going through that. So this pandemic is a new disease. I write the keywords. Say for example, a new disease which spreads over a vast geographical area. So it spreads over a vast geographical area covering many countries and continents, and the people have no immunity against it. So all the terms are relatable to coronavirus disease. No immunity against the disease, and when this kind of disease occurs is known as pandemic. So it's a new disease. Most of the people, like 99% of the people, do not have immunity against it. It occurs in a vast geographical area. This is known as pandemic. Other diseases which has been called pandemic earlier is Spanish flu, cholera, it's influenza, etc. Now what is epidemic? Epidemic disease is, it is a disease which affects again a large number of people within a community. Within a community. Or within a particular area or a population. Okay, now endemic is what? It is the disease that belongs to a particular people or a particular country, specific to a particular country or a particular group of people. And The sporadic disease means they are scattered disease. Haphazardly they are distributed. Say for example polio, meningococcal, meningitis, tetanus, etc. So these diseases are scattered about. Clear? So here the question was, when a disease occurs periodically in a widespread area causing devastating damage to a crop plant, it is known as epidemic disease. Question number 21 is disease caused by zinc deficiency. Now this is a very common question which is asked very frequently. So the disease caused by zinc deficiency, it is Khaira disease. Next is radicle is enclosed in a protective cover. In the last question we did, the radicle, that is the root in monocots, is enclosed in a protective cover, coleorhiza. When it is about shoot, then it will be coleoptile. Very easy, no? Now, an antibody present in blood serum specific to an antigen present on red blood cells is called as. Now, an antibody present in blood serum which is specific to the antigen present on red blood cells is called as agglutinin. So, antibody which is present in the bloodstream, and this antibody, it is specific to the antigen on the red blood cells. Now every blood cells or every cells expresses an antigen on their surface, okay, and to the antigen antibody binds. So this antibody which binds to the antigen on the red blood cells is known as agglutinin. The next question is the link between the vertebrates and invertebrates. The link is known as amphioxus. The bacterial cells divide mainly by a process where a single nuclear division followed by a division of the cytoplasm, first the nuclear division then the cytoplasmic division, to form the two daughter cells of equal size is known as, this is known as binary fission, mainly seen in bacterial replication, where the nuclei first will become two and then there will be the cytoplasmic division forming two daughter cells. Okay, so this is binary fission. Next question is number 26. The condition that refers to the absence of sensation of smell, it will be C, which is anosmia, which is mainly we see when there is like the symptoms of COVID is that the person loses the sensation of smell, anosmia. Okay, but in COVID there is the temporary loss of the sensation of smell and taste. Next is cell-to-cell communication in legume rhizobium symbiosis is carried out by, this we studied in nitrogen metabolism in plants, how symbiotic fixation in plant occurs with the help of rhizobium bacteria. Now this is by the lectins. Okay, these lectins, they are the sugar binding proteins, and these lectins are expressed on the surface of the plant cells. Okay, so this is helping in the cell to cell communication between the rhizobium bacteria and the leguminous plants, right? The on the surface of the leguminous plants there will be the lectins which will be recognized by the rhizobial bacteria. Next question is diseases caused in plants other than a living organism. So if it's living organism, it should be biotic. If it's other than living organism, it will be abiotic disease. Endemic disease we already discussed, that a disease which occurs periodically in a widespread area. Okay, next is, so epiphytotic disease, if we discuss about this epiphytotic disease, is that whenever the number of individual a disease affects increases dramatically, means the number of individuals which the disease affects increases dramatically. Number of individuals, just a moment. So, number of individuals increases dramatically. Since it is related to plants, so it is epiphytotic. If it would have been related to animals, it would have been epizootic. Dramatically. Okay, I hope you all are understanding. Our next question is number 29. The short chemically synthesized DNA double strand, which can be used to link the ends of the two DNA molecules having different sequences at their ends when used in conjunction with the linkers. So these are used in conjunction with the linkers, and they are used, for example, they are used to link the two DNAs together. Okay, so these are short and they are chemically synthesized. They are not naturally available. They are chemically synthesized double stranded DNA molecules, and they are used to link the DNA having different sequences at their ends. Okay, if these they have different sequences at their ends, then these adapter molecules are designed to link the DNA together. Okay, these are also double stranded. They are designed to link the double stranded together. These are adapters. Number 30, sunflower crop is also known as indicator plant for the deficiency of. So sunflower indicates the mineral nutrient boron. Is it a micronutrient or macronutrient? If you remember, yes, this is micronutrient, means required in small quantities by the plant. And sunflower crop are the best indicators for for boron. Okay, next is hot water treatment is used to break dormancy in. So lentils dormancy are broken by hot water treatment. Next is nematicides. Nematicides means the ones which can kill nematodes. They inhibit the enzyme of nervous system in killing of nematodes. Which enzyme do they inhibit? Which enzyme involved in nervous system is inhibited by the nematicides? So nematicides is a kind of pesticides which inhibit an enzyme involved in nervous system of the nematode. So when this enzyme of the nervous system of nematode is inhibited, then nematode dies. So which enzyme is that? It is acetylcholine esterase, very important enzyme. If this is a neurotransmitter, if this is inhibited, nematodes are killed. The signal will not be transmitted to the brain, right? Next, number 33, sulfur containing amino acid. I think this you all know, guys. Cysteine and methionine are the two sulfur containing amino acids.